Bismillahirrahmanirrahim. Assalamu alaikum friends. In this video tutorial we learned that enzyme involved in DNA replication. So very simple topic, and I explain very easily this topic. So now coming to start the topic, that is basically enzyme involved in DNA replication. So we know that before the process of replication, or before the cell division, DNA occur in nucleus, so in the form of coiled or spherical palm. So when the process of replication are started, the DNA are converted into leader shape. So what enzyme are involved in this process? So the first enzyme which are involved in the DNA replication is topoisomerase or gyrase, topoisomerase 1. So the first enzyme is basically Gyrase. Or topoisomerase 1. So what is the function of gyrase or topoisomerase? So we explained that before the process of replication, the DNA are found inside the nucleus. So they in the form of coil shape or spherical palm. So in this palm, DNA are occur inside the nucleus. When the process of replication are started, so the coil palm of this DNA molecule are converted into leader shape. So what enzyme are involved in this process? So basically the enzyme which are involved in this process is gyrase, which convert the coil palm or the spherical palm or the twisted palm of DNA into leader shape. So enzyme which are involved in this process is basically gyrase. So gyrase convert this structure of DNA into leader palm. We know that in two polynucleotide chain, the hydrogen bond are present, which are adenine attached with thymine and double hydrogen bond, and cytosine and guanine attached with the triple hydrogen bond. So when gyrase perform its activity, so the second enzyme which involved in DNA replication is basically DNA helicase. So what is the function of DNA helicase? When this coil palm of DNA are converted into leader shapes, so the helicase enzyme are attached in this position in this hydrogen bond, and denature the hydrogen bond to form two template strand, which are synthesized for new strand. So the function of helicase enzyme to break the hydrogen bond which are present between the two polynucleotide chain. So this structure are converted into this palm. And it's continue its activity in the replication process. So this is a break due to helicase enzyme. Now the third enzyme which are involved in DNA replication is basically protein, single strand binding protein, single strand binding protein. So what is the function of this single strand binding protein, or we simply call S single strand binding SSBP. So what is the function of this single strand binding protein? It provoke to reattach to or rebinding this hydrogen bond, or to form this hydrogen bond. It's prevent formation of this hydrogen bond again. So it's attached on these side of DNA molecule. So it provoke the rebinding of or formation of hydrogen bonding. So now the main enzyme which are involved in the DNA replication is basically the DNA polymerase. So main enzyme in the process of DNA replication, it's synthesize the new strand or a daughter molecule of DNA. So it's have main three types in prokaryote. The first one is DNA polymerase 1, second one is DNA polymerase 2, and the third one is DNA polymerase 3. So what is the function of DNA polymerase 1? So it's do its function in the termination phase of replication. Do its function in termination step of DNA replication. Now the second enzyme, which is the type of DNA polymerase. So what is the function of DNA polymerase 2? It's repair the daughter molecule of DNA, and also perform its function when mismatching occur in the new strand. Basically, mismatching is a phenomena in which we know that nitrogenous base is basically adenine, thymine, cytosine, and guanine. When we know that adenine, I want to draw the diagram of DNA molecule. So this is the one strand and this is the another strand. This is the A and this is the T. We know that adenine match with thymine and cytosine match with the guanine. When mismatching occur in the daughter strand or a new strand of DNA molecule, so it's perform its activity when adenine match with cytosine. So it is error occur in the DNA molecule. So it's do its function to prepare or to remove this unfair of cytosine molecule and replace this cytosine molecule into thymine molecule. So that is the function of DNA polymerase second. And the third one is DNA polymerase 3. So DNA polymerase 3 is the main enzyme of DNA polymerase. So it's synthesize new strand, it's add nucleotide into new strand. So add nucleotide into new synthesized strand. Now the fourth enzyme which are involved in the process of DNA replication. That is basically primase. We know that DNA molecule are DNA polymerase enzyme do not work without primer. So primase enzyme are those enzyme which make RNA primer for the DNA polymerase enzyme. So it's make primer for DNA polymerase to do its function. Without primer, DNA polymerase not do its function, so it is the drawback of DNA polymerase enzyme. Now the fifth enzyme which are involved in the process of DNA replication, basically gyrase. Ligase. So ligase are also known as molecular glue. So what is the function of ligase enzyme? So it's bind the two nucleotide strand of lagging strand. We know that it's covered in the when I upload a second lecture on DNA replication. So inshallah the function of ligase enzyme are covered in the next lecture. So I want to shortly explain this work of ligase enzyme. So this is the lagging strand or discontinuous strand. Which are synthesized discontinuously. So there are a short fragment which is known as basically Okazaki fragment. So when it's removed from this side with the help of enzyme DNA polymerase 1, it's do its function in the termination phase. So when remove the Okazaki fragment from these side, when it's removed, so and the DNA polymerase 1 attached attached to nucleotide in this area, so there are some space are occurred between the two nucleotide molecule. So ligase join and make phosphodiester linkage between this nucleotide and this nucleotide. It's make phosphodiester linkage between two nucleotide. So this is the overall enzyme which are involved in the process of DNA replication. So if you are new in this channel and learn something from me, so please subscribe my channel, share and like. Thank you. See you in next time.